Uh, um, so welcome to the second part. In the first part, we stopped at radial cleavage. So for the second part here now, we want to start from the spiral cleavage. And if you look at the diagram, I think the diagram has a way of explaining more about it. And like I wouldn't want you to so much put all those diagrams and all those stuff. You don't really need to put them so much in mind or whatever. Just try to understand the type of cleavages we have, and try to know the definition and the diagram. Nobody will ask you to draw the diagram, but when they say something is spiral, so you can as well draw a circle, like, okay, like in this diagram, for example. Now this is showing you that it is spiral compared to the first one that we did, this radial. You can see that for radial, all the line of planes, all those line, they are straight. If you see the way the cursor is, everything passes through the center for radial. But when you come to spiral, you will notice that it does not pass through. The first arrangement, which is the basement, the second one that has been arranged on it, arranged itself at the center of the one below. And the next one again at the center, the next one again, like that, like that. So that is the spiral part. So the plane is inclined to the long axis of the embryo so that the smaller one, the macro may lie over the intercell boundary between the macromeres, which is what I just finished explaining. So spiral cleavage equals unequal holoblastic. It will be unequal. Now, each cell can be recognized up to the 64 cell stage. Now, most of the ones we talk about, I think we talk about 8 and I think 16 and so on and so forth. So here now we are saying for spiral cleavages, you can have up to 64 cell making up the spiral cleavages. Also, the shortest line drawn from one pole to the other without cutting through the side of any cell must be spiral. I think you understand this. Aha. So it is a determinant or determinate type blastome with fixed fate and easily seen in embryos of protostoma coelomate, e.g. in marine polychaetes, that's annelids and mollusca. So all we're just trying to say, like I said, um, different organisms have different cleavages, and that is because of the type of animal that is going to be formed. Now, when you look at the previous one, when we're talking about the radial, you see that we're talking about echinoderms. And normally, echinoderms are what? Are radially symmetrical. If you look at starfish, for example, now you see the way it is. So every angle you cut it, it will always have a center. They will always have a center. But when you get to this spiral now, which is annelids, I think from this very one, you will have a better understanding in earthworm. If you look at how earthworm looks like, so this is also an example how it is being arranged before you get to the last one, and also mollusk. So that is all about spiral cleavage. Like I told you, just try to know the type of cleavages and try to have a better understanding on how to explain them. So talk about the plane, talk about um talk about the number of cells that make them up. So those are the main things that you will have. And also in short answer questions, you can see something like that there. The cleavage that has this and this and that. So those are the questions you will likely see. But definitely when we get to the question and answer part, I'll try as much as possible to drop some questions so you can use that to know how the question is being asked. Now the next one is superficial or endothelial cleavage. Now, like I said, the reason why I always try to show you the picture first, so that when you are reading the information, you quickly get the idea behind it. Now, if you look at what we have here, we have yolk here, and here is what's it called? Here is like the nucleus. It's one side, and it gets to a particular stage where you have three nuclear stages all around. And after a particular stage, after another stage, you see that the nuclei are now arranged. If you look at this very structure that we have here now, you'll be able to remember when we discuss when we talk about blastula, if you remember very well. So which means there's an advancement already in that kind in the organism that belongs to that stage. There's an advancement compared to the other ones we've been talking about. Now, this is the characteristics of yolky centrolecital eggs of arthropods, especially insects. Mitotic division of the single nucleus, the one nucleus that is here, undergo mitosis. When we get to cell division, we'll talk about mitosis and meiosis. So you'll be able to differentiate between mitosis and meiosis, which I know you would have definitely have the idea. So mitotic division means the one cell multiplied to two, but here now we're talking about the mitotic division of the nucleus. One nucleus turned to two to four to eight and so on and so forth. So the mitotic division of the single nucleus results in the migration of several nuclei from the central yolky mass, to the peripheral, which is relatively yolky free, to all the sides, which is what? Yolky free. And that is what you have here. Cell wall are then formed around them. Cell wall are then formed around them. So the yolk will be in the center, while you will have what? The nucleus all around, forming the base of what? Of the tissue. Now you will discover when I showed you the diagram of epithelial tissue, you will see that in between every every tissue you have cells there. So this is just an example of what we are trying to say there. So the next one, during rapid division of the fertilized egg, a spherical mass of cells, which is called morula, is formed. Now, as process continue, the cell draw away from the center, leaving a fluid-filled cavity. I think I've explained all that from the picture here. This multicellular complex is now called blastula. If you remember, I went back to show you that. Individual cell is called blastomeres. Individual cell, each of those cells are called blastomeres. And the cavity as blastocoel. The fluid-filled cavity inside is called what? Blastocoel. So the blastula of centrolecital eggs lack blastocoel. So which means the one with centrolecital egg, if you remember centrolecital eggs, center, yolky egg, they said it lacks what? Blastocoel. Now, if you want to get that, you go back to when we discussed about this centrolecital egg. I made mention of that. The reason why it will lack blastocoel. So please let's try to get that, because these are the short answer questions that you might likely see in the short answer question part. You can just say the blastula of centrolecital egg lack dash. Very simple. So you just have to know it from what we have here. And that is why I decided to make it what's it called, bold, so that you always be familiar with them. So we're going to the next one, which is the partial or meroblastic cleavage. Now, in eggs with large quantity of yolk now, meroblastic cleavage, grooves are confined to the cytoplasmic blastodisc because the cleavage is inhibited completely in the yolky parts. Now, at the yolky part where we have more yolk, that is where you have more cleavages occurring. So if it is at the upper part, for example, now you will see a lot of cell division going on there, or cleavages going on there, forming all this. So you can have the other part below. Completely in the yolky part, this type of cleavage is known as partial or meroblastic, and it contrasts with total or holoblastic cleavage. So the diagram has actually explained it. So it's meriblastic or partial. It occurs. That is just the simple English. The grooves are confined to the cytoplasmic blastodisc. Why? Because the cleavages is inhibited completely in the Yoki part. The Yoki part, that is where you have what's it called, the cleavages occurring. So some part will be free where you have the Yoki part going through what division. So those are the type of cleavages that we have. So just try to know the type of cleavages, know what's it called, how each of them works, so that when question comes on them, you know what to do. So the next one we're going to gastrulation. Gastrulation. It means formation of gut. It means formation of gut. Method of gastrulation varies among animal species, but it is the invagination or inpushing of one end of the blastula. I hope you still remember all those diagrams. I think the next page will still talk about it. I mentioned it, although it's more like an introduction to it then. So, invagination or inpushing at one end of the blastula. One end of the blastula we what's it called? Invaginate, inpush. It will be pushed in. Okay, e.g. in amphiosis, cells of vegetal pole. If you remember the vegetal pole, begin to roll inward. The vegetal pole is the lower part, while the animal pole is the upper part. So the cells are the lower part. That's because that is where we have the yolky part. So it begins to roll inward. And as it is rolling inward, it is forming a depression and later form a two-layered cup-shaped gastrula. Now, before I continue with the other one, let me just quickly show you the diagram here. Now, this is the blastocyst. Blastocyst, if you remember, I said the fluid-filled cavity part. So that is from blastula. For blastula, you just notice that what cells and the nucleus begin to form all around, and in the center you have the fluid-filled blastocyst. Now, um, as it continues, you will notice that what the cells at the lower part, which is the vegetal part, begins to it begins to draw itself inward. It begins to go inward, as you can see from the diagram. It begins to go inward, and as it is going inward, it is going to form a false gut. Because I call it a false gut, but it's also called a primitive gut. Primitive meaning something that is not the real gut. It's not the real. A gut is more like when we talk about esophagus. So it's not the real gut, but that is where gut also starts imaginating from. So we call it primitive gut. So you see the invagination going in, and after some time, you will see the opening, which is the blastopore. You have the endoderm, that's internal, and you have the ectoderm, which is the external. Now, that is for early gastrula. But as the invagination continue, it will now later form the three germ layers. The three germ layers are now the mesoderm, the ectoderm, and the endoderm. You will really appreciate these things very well when we get to animal diversity, because that is how we're able to differentiate between all the animal kingdoms that we have. Yeah, the jam cell, the jam layers, it really explains the differences between cells. So you are going to appreciate it more when we get to animal diversity. But just have the understanding here that once invagination occur, while gastrulation is taking place, and there's invagination at one end, which is the vegetal part, and there's invagination at the vegetal part, and you have the archenteron being formed, you have the blastospore being formed. The first two layer that you first have is the ectoderm and the endoderm, and finally the mesoderm will join. The mesoderm basically is formed um from the blastocyst, because in some animal it first come up in form of a mesogly, or mesoglea. So it's also a fluid-filled part. But in higher animals, the mesoglea or the blastocyst become solidified. Then you call it the mesoderm. We'll still talk more on that. Don't worry, you really understand better. But just have the understanding now that the first two parts that will be formed is ectoderm and endoderm, in which you have the fluid field in the center that will actually form the acantharon. And after a while, it closes up, then you have the mesoderm. So let's go back to where we are. So now here I said that deeper laying cells form endoderm and mesoderm. Deeper laying form endoderm inside and the mesoderm. Why the outer layer form the ectoderm? I just finished explaining that. Now, so gastrulation can then be defined as the process that results in germ layers. So what are the germ layers we have? We have three germ layers, which is ectoderm, endoderm, and mesoderm. So those are the three germ layers that we have here. That is the cell layers from which tissues and organs of adults arises. If you remember the diagram I showed you from the beginning, let me quickly go back to the beginning so that you will appreciate this better. Now, if you remember this, I said the organogenesis. Organs are formed from this ectoderm, mesoderm, and endoderm. So that's the same thing I made mention of now. That's the same thing I just made mention of here now. That gastrulation can then be defined as the process that results in germ layers. That's the ectoderm, endoderm, and mesoderm. That is the cell layer which form the tissues and organs of adults. That's where they arise from. Note that organs do not arise directly from the cleavage cells. I think you should understand that now with my explanation. The organs do not arise directly from the cells formed during cleavages. The cells formed during cleavages, basically, they form tissue. So after they now form tissue, then tissue will now begin to arrange themselves. It will begin to arrange themselves in the order it's supposed to arrange. Then tissues that have the same characteristics, that have the same function, they now come together. That is when you now have the mesoderm, ectoderm, and the endoderm. Then they will now begin to form your organ. Remember, organs are formed when specialized tissues come together. So that is that about that. So to the next page now, still talking about the explanation we have there. So gastrulation also converts blastula into a gastrula. From the beginning, I've explained this, how blastula get into gastrula. And the same thing is what we have here. It's all started from blastula and ended with what? Gastrula. And blastula, if you still remember, you have cells forming around with the nucleus inside. That is blastula. Immediately invagination begins to occur, blastula is being changed into what? Gastrula. And how do you know that gastrula has been formed? That is when you now have your ectoderm, mesoderm, and your what? Endoderm. So during intalking, I think I've explained all this, which is from what we have here. Immediately there's intalking, there's a new cavity, which is the acantharon, which is also called primitive gut. This contributes to the formation of the gut cavity. Now, the opening of the acantharon to the exterior where invagination occurs is the blastospore, which is like the opening. Now, in deuterostomes, the blastospore becomes the anus. I mentioned that if you had listened from the beginning of this class, that is the blastospore that actually form the mouth and the anus. So it becomes the anus of the fundamental larva and often of the adult, while the mouth will be formed as a result of ectodermal invagination. So blastopore basically will form the anus, which is the lower part. Now, more invagination of the same ectoderm what's it called? Ectodermal, more invagination of the ectoderm will now result into the mouth. So it's not just this one opening forming the two. But in early organisms, for example, when you talk about Nigerians, for example, now they have only one opening, and that one opening serve as the anus and as the mouth. Okay, so that's that. In protosome, blastospore forms the mouth. Ha, that's just what I finished explaining now. In protosome, the blastospore forms the mouth. Blastospore in insects is a furrow through which a ventral strip of a prospective mesoderm cell is invaginated. Now, as organisms are advancing, are you getting me? The mesoderm also gets involved in the formation of the mouth and the anus. But basically, most anuses are from this blastospore. So the next part now. So for the next part here, that's with the three primary germ layer, I've been mentioning that since, cells regroup, rearrange into cell masses that give rise to tissue. If you, my explanation has talked about that. So the three germ layers are endoderm, mesoderm, and ectoderm. I've explained that. Now the germ layers are then differentiated because once this ectoderm, mesoderm has been formed, that is more like the tissue level of organization, tissue level organization. So from tissue level organization now, then you now go into the formation of organ. So germ layers are then differentiated into 5 major tissues by a process called histogenesis. Histogenesis, more like study of histology. So which are the epithelial tissue, the nervous tissue, the connective and supportive tissue, the vascular tissue, and the muscular tissue. Now that is what I said earlier, that this histology, which is or histogenesis, is the study of tissue. So I said we will not really go much into it because it is a whole topic on its own. So and it has its own full what's it called, syllabus, which we are going to deal with all these 5 tissues in the body. So we'll talk about them one after the other. Now, the cell masses continue to differentiate to form organ and system, which is called organogamy. So I told you that also. We will not be going into the study of organ because we'll still talk about all the organs too. So at the end of gastrulation equals complete enclosure of both the endoderm and mesoderm by the ectoderm. So from the previous diagram, you would have seen that. So all these ones too, I think I've given you the expansion from the beginning, but I'll still just read it out so you can get that. So ectoderm give rise to skin, nervous system, and associated structures. Mesoderm to the supporting and muscular structures with circulatory um, or vascular tissues. The endoderm, the liver, the pancreas, the digestive tube, and so on and so forth. So all this, what we are trying to say here is the 3 of them leads to the next stage. That's from tissue to organ. So you will see them coming together as tissue to now form a particular organ in the body. So in most triploblastic are animals. They are animals that have the 3 germ layers. Now, I'm mentioning all these things and making emphasis on them so that you'll be familiar with them. So when we get to animal diversity, it will be very, very easy for you. If I ask you to give me 30 characteristics of Annelida or whatever, you'll be able to get it because you have the foundation of how this whole thing comes in place. So when you hear the word triploblastic, so we tell you, you will say, okay, annelids are triploblastic animals. So why do we say they are triploblastic animals? Because they have the ectoderm, mesoderm, and the what? And the endoderm. So, and a cavity, which is the cilium, is formed within the mesoderm. Now, this cilium, we'll talk about it properly, and that cilium actually results to the full formation of organ level. But we'll talk about it. We'll talk about it We'll talk about it. So cilium is more of a fluid-filled cavity where you have the development of organs. So that starts from triploblastic animals, which is what annelida. So when we get there, we'll talk more about it. In some coelomates, the protostomia, the mesoderm splits, leaving a cavity which enlarges to form cilium. This mode of cilium formation is called schizocoely. Schizocoely. So they are trying to talk about a particular type of coelomate animal, which is a protostomy. It is still called cilium, but their own cilium is called schizocoely. So just take note of that. These are short answer questions that they can ask you. So they can say cilium is called dash in protostomia coelomate. So you should be able to have this. I've come across it in IGMB question many times. So try to get that. So the mesoderm in some other coelomates, that's deuterostomia, like I said, we will still talk about all this, so you'll be familiar with them when we get there, is formed by the wall of the achentheron. I think you still remember that. That's the wall of achentheron form the mesoderm, invaginating to form pouches, which then separate from the achentheron. So the pouches lead to the formation of their own coelom. So they are just trying to tell you that in different animals, it has some other names, and also it can be formed from the neighboring cell, and that's why they are trying to mention this deuterosomia. Now, the cavity and the wall become the coelom and mesoderm respectively. This is called enterocoely. So they are trying to tell you how the coelom come in place. The cavity of the wall cavity becomes the coelom. So we already know about the coelom, the fluid-filled cavity, and the mesoderm respectively. So together, it is called what? Enterocoely. Now, the outer layer of the mesoderm adjacent to the ectoderm is now called the somatic mesoderm. Well, like I said, we don't really need to wrap our head so much on most of these things. Just get the information, which is more like saying, in coelomates, that's protostomia, the mode of coelom formation is called dash. Just register it. Just register it. In this one, when coelom and mesoderm are formed respectively from the cavity, it is called dash. Wrap it around your head. And the outer layer of mesoderm adjacent to the ectoderm is called dash. They are always short answer question in IGMB. So when I bring past question and I want to start treating past questions with you online, you will understand what I'm trying to say. It's just short answer question. They just want you to fill it in. Just fill it in and you are good to go. Thank you. And we go to the next one. Still trying to bring out questions from short answer question from this topic. So the inner layer of the mesoderm adjacent to the endoderm is called the splanchnic mesoderm. Inner layer of your mesoderm. Mesoderm is the inner layer inside. So the inner layer of the mesoderm, which is adjacent to the endoderm. Don't forget, mesoderm and endoderm, they are internal. Ectoderm is outside. So the inner layer of this mesoderm that's adjacent to endoderm is called this. Take note of that. The previous one says the outer layer of the mesoderm adjacent to the ectoderm is called this. So they want to ask all those questions and they want to confuse you with this. That is why I decided to put it as points so that you can take note of them. Here is outer layer of the mesoderm adjacent to the ectoderm. Ectoderm is outside. Is the one that form the outer layer. Now they now say outer layer of the mesoderm. Mesoderm is in the middle. So outer layer of the mesoderm, which is now adjacent to the ectoderm, that will give you the somatic mesoderm. Now the inner layer now of the mesoderm, which is adjacent to the endoderm that is also inside, is called splanchnic mesoderm. So is that. Now, somatic, that's the first one we talked about. The somatic mesoderm, if you remember the somatic mesoderm now, which we talked earlier, before we talk about this, So the somatic mesoderm plus the ectoderm equals the body wall. The splanchnic mesoderm plus the endoderm equals the wall of the digestive tube. So, like I said, try as much as possible. After viewing this on the YouTube page, make sure you download it. Make sure you download it. And the funny part about online classes, it can be very, very sweet. Just look for your headphone or your headpiece. At night, if you want to sleep, just keep listening to it and just jot it down. Carry your past question and start solving. So you don't need to start looking at your book and grammar, and you are feeling sleepy. Even you can be walking on the road. Just make sure it's a safe place. Just plug your earpiece and be listening to this thing I'm saying. So by the time you begin to listen to it, you see that it's going to sink. That's something about online class. And that's the reason why we are trying to bring online class in, plus the physical class, so that both of them will make you develop yourself, both from hearing, online, and also the physical class. So the other ones, I just wanted to go through it. So a longitudinal support rod, the notochord, is formed from the mesoderm, and just above the notochord, the ectoderm becomes thickened to form a neural plate. Now, all this now, we are now trying to move into a more developed organism, because now it's like we are leaving the we are going to a more complex triploblastic now, where the neural plates now begin to come in. All those things you are appreciating more when we do what's it called, animal diversity, because we will explain them more based on the animals that have them as organisms are developing. So the edges of these plates approach one another to form the neural groove, and then continue until the fuse in the mid-dorsal line forms a dorsal, holoneural tube, which is covered by ectoderm. The neural tube now gives rise to most of the nervous system. Like I told you, when you begin to talk about neural plates, you begin to talk about the neural groove, then it means you are already in chordata, in the chordates. Are you getting me? You're already in the chordates, echinodermatas and echinoderms and the chordates, which where we have the most developed organisms. So then you are now talking about you are talking about organ system level. Like we belong to organ system level, because we have brain separately, we have the digestive system separately, we have the excretory system separately. But all those the early organisms, they don't have organs. Most of them don't have organs. They belong to the tissue level of organization. The organ level of organization actually start from Annelida, and that is because it has a digestive system which run through. But as you go higher, you now begin to mention the neural plate, the neural groove, the neural tube, where you now begin to talk about the spinal cord. You have nerves running around, and that is where we come across the nervous system coming in now. Its anterior end enlarged to form the brain, the cranial nerve, and eyes, while its posterior end form the spinal cord and the spinal motor nerves. The special neural crest cell cuts off from the neural tube before it closes, giving rise to the sensory nerves. Well, like I said, this is not to be really discussed here, but we are just trying to tell you how the nervous system come in place, the spinal cord, and every other one, the sensory nerves, how they were able to develop. So we are all still trying to tell you that they all develop from the ectoderm, the mesoderm, and the endoderm. So that is the reason why you have them all here. But try to take note of them so that when we get to animal diversity, you will quickly understand where we are going to about them there. Okay, thank you. And I think this will be the last topic under this, which is the development of birds and mammals. Development of birds and mammals differ from that of amphibians and frog in two main ways. So remember, we've been talking about every of the how neural what's it called um the ectoderm, the mesoderm, and the endoderm, how they come in place, how they were able to link together and form the nerves, the nervous system, and so on and so forth. So we're not trying to look at the development of birds. For those of you that are kind of familiar with biology or biology question, you would have seen this very diagram. This very diagram. I know you would have come across it in some of your past questions because they want to ask one or two questions there, even in what's it called, even in practical, they will just bring it and ask you to label it. So we'll come to the diagram. But before then, let's look at how birds and mammal differ from that of frogs and amphibians. Now, egg of birds contain much more yolk, with the result that cleavage is only confined to the animal whole. That is, embryo is situated on top of a yolk sac from which it derives its nutrients or nourishment. So that is why they said the development of birds and mammals is a little bit different from frogs and amphibians, so that you have the idea. So what makes it different is egg of bird. It contains much more yolk, with the result that the cleavage is only confined to the animal pole. Remember most of the explanation we gave, the telolecita and so on and so forth, you will have more yolk at the vegetal part, not the animal pole. But for this one now, you have it's mainly confined to the animal pole. That is, the embryo is situated on top of a yolk sac. And the reason why it's there is because that is where it derives its own nourishment. Embryos of birds and mammals are surrounded by a system of protective membrane. It is surrounded by a system of what? Protective membrane, which develop from tissue outside the embryo itself. These are known as the extra-embryonic membrane due to its origin. So let's go to the next one. We still have a lot to discuss under the diagram. Now, most times when IGMB or JUPEB asks questions on this thing, they draw this diagram, they want you to label it, and after labeling it, they want you to tell them the function of each of the parts labeled. Now, so we want to just look at them. There are three other embryonic membranous sac in addition to the yolk sac, namely. Now that's the yolk sac. You can see the diagram here. You can see the chorion. You can see the allantois. You can see the amnion. You can see the embryo itself developing. You can see the amniotic cavity within the amniotic fluid. You can see the shell protecting the egg. You can see the albumen, and you can see the yolk, which is the nutrients. So let's take them one after the other and talk about them so you'll be familiar with them. So the first one is the chorion. That is the outermost extra embryonic membrane, which encloses others. The outermost extra embryonic membrane, as you can see, it encloses others. Everything that is inside, it encloses it. That is the chorion. Please, all these definitions, be very familiar with them and make sure you are familiar with this diagram so that when you see questions like this, to label it and to explain the function of the part, so you'll be able to know what to write. So the next one is the amnion, as you can see this diagram of the amnion, it is a fluid-filled sac that encloses the embryo. As you can see, the fluid-filled, it has fluid inside, so that the embryo will not go through shock and physical disturbance, to avoid shock, as you can see. So it is a fluid-filled sac that encloses the embryo and protects it from shock and physical disturbance. That is the what? The amnion. The amnion. Please take note of that. Remember chorion, remember amnion now. So the next one is the allantois. Allantois. First look at the diagram. As you can see it here. Good. It's a sac that grows out of the end gut of the embryo and collects its metabolic wastes, mostly uric acid, in births. So it grows out of the end gut, the gut behind. End gut is more like talking about the anus, something like that. So, but basically you don't call it anus here. That's because the metabolic waste being collected is mostly uric acid. So it is not like it's really solid. So the wastes from the embryo, newly developing bed now, the wastes from them is basically uric acid. So it is the function of the allantois to collect all those wastes. So we go to the next one. Now, the chorion and allantois. The chorion, as you can see, which we've mentioned already, and the allantois, as you can see, there's a part where the two of them join together, as you can see here. So they fuse together to form a chorioallantoic membrane. Chorioallantoic membrane, located just beneath the shell, and provided with rich network of blood vessels, connected to the embryonic circulatory system. So, what is the function? Why do the two of them now come together to form a relationship? So that they will act as the circulatory system for the developing embryo. It's as simple as that, because they have rich network of blood vessels. Now, this allows free exchange of oxygen and carbon dioxide to occur between the blood of the embryo and the surrounding atmosphere. God is great. So that is the function to that. And as the embryo of reptile, birds, and mammals develop within an amniotic membrane, these animals are referred to as amniotes. Amniotes, which is from the word amniotic. So when animals that develop within an amniotic membrane, these animals are referred to as amniotes. So all this very one now, it's an example of asking a short answer question. Animals that develop from within the amniotic membrane are called dash. So take note of that, because when my tests begin to come, these are some of those things that you see, and that is why I decided to highlight them so that you be aware of them anytime you see questions on them. And the next one here, which is I think it should be the last slide. So the embryo of fish and amphibians are without an amnion and therefore are called anamniotes. Now, anamniotes, an means none. So anytime you see an in front of a word, it means they don't have it. And they are none, because they are without an amnion. Now, during development in advanced placenta, now we are going to mammals now. Mammals, including man himself, a foetal maternal structure develops. that is more complex to that of birth now. So what does it do? It protects mammalian embryo by being highly selective, and it permits the entry of foodstuff, hormones, vitamins, and oxygen, and the removal of carbon dioxide and metabolic waste. Remember that. That is placenta, advanced placenta now. That is in mammals, because all this chorion, all these ones cannot do the work again. So we need something more advanced. And that is where it comes in as placenta. So when we get to more of reproductive this thing and other stuff, we'll still talk about placentas and co. So as the allantois, if you still remember the function, as the allantois is no longer needed for excretory purpose, because in human now you are developing a big and well-grown human, which is bigger than what you have in eggs. So in eggs now you still have what allantois to remove excretory products, but in mammals now you don't need allantois again. Instead of allantois, you now have a well-developed one, which is called placenta. So now it becomes the umbilical cord that link the embryo to the placenta. So we don't need the allantois, because it will become the umbilical cord that will now link to the uh placenta, link to the placenta. So the outermost membrane, that's the chorion, forms most of the placenta, the amnion is still fluid-filled and unchanged in function. So that's all about this. Please make sure you know the diagram, be very familiar with the diagrams and the labelings, basically. Be familiar with these labelings and the function of each of this. You know what the shell is for, the albumen, the yolk inside. We talk about that, the chorion. But basically 1, 2, 3, because we are really talking about the reproductive part there. So the main functions, that is what we have given here. So thank you very much. And please, like I said, make sure you download, make sure you subscribe. Try to subscribe. I think that's the best way. So if you subscribe, once we drop a new class, you'll be able to know, and you can just start working there. And if you also subscribe, um even when you finish your program and you leave, when you are in school, you can always go back there and watch the videos, because it's really going to help you. And I don't know, because it's like I said, we are spending extra money, and the physics and the chemistry class, the mathematics class also will be starting soon, because the teachers also are working on their own. So very soon it will be starting, so we wouldn't have any issue at all. So once we have all the classes going on, it's really going to help you. So you are doing the physical and you are doing the online. So thank you very, very much.